Hi, good afternoon. I'm Pevindu Abeysinghe. I'm from Queensland University of Technology. I'm a PhD student and I'm working at Center for Children's Health Research, School of Biomedical Sciences. And let me share my screen for our presentation. I hope everyone can see my presentation. All right. So my presentation will be exosomes derived from microRNA and how they can be used as a diagnostic and therapeutics. And let me start it. So what are these exosomes? These exosomes are pretty much membrane-bound vesicles. They're pretty much like Golgi bodies or endoplasmic reticulum in the cell. And these exosomes are a bit different from Golgi body or any other vesicles because it contains many important and critical biomolecules, for example, proteins or RNA molecules or DNA or lipid molecules. They all can be engulfed into one of these membrane-bound vesicles, exosomes, and they can be transported from cell to cell. That's the most important thing here. So they act as carriers or vehicles of these biomolecules from cell to cell. And the other important part is these are pretty much small vesicles. They size from 30 to 150 nanometers. And so as in here, these proteins and all these important biomolecules, they engulf into endosomes, which are the initial types of exosomes in its biogenesis. And after that, they will be shed out into the intercellular space from the cells. And there we call the exosomes. So these are pretty much novel and very important class of biovesicles. So I would say there are many more functions in regards to these exosomes. For example, they transduct signals from cell to cell. They contain many important genetic materials, for example, DNA and even RNA and some other types of RNAs, for example, small RNA types and longer RNA types. And these exosomes are involved in epigenetic modifications and even in immune system regulation. So these are pretty much few of these functions of exosomes. But I would just move on to the important or my expertise, which is exosomal microRNA. I'm more interested on these microRNAs, which are engulfed in these exosomes, because so these microRNAs are pretty much small RNA molecules. So there are some RNA types, rRNAs, ribosomal RNAs, tRNAs, and even messenger RNAs. So apart from these types of RNAs, these microRNAs are pretty much small RNA molecules, yet they're pretty much important in gene expression. And the other thing is these microRNA molecules are selectively packaged into these exosomes. So they are not haphazardly packaged in. They depend on their origin. For example, the production of a microRNA would start from the nucleus. So it will start off as a precursor microRNA, and it will then transport into the intercellular space, and it will develop into a mature microRNA, and finally they will be like engulfed into endosomes, and endosomes will be developed into exosomes. So these exosomes release into this extracellular environment, and then these recipient cells, another different cells. This cell may be few kilometers away from your body, like the origin. So for example, if the exosome originates from a heart, and they can be transported through the blood vessels, and they can be reached into our into a reproduction organ, or into our brain, or the recipient cell may be a different type of a cell when compared to its origin. And these microRNAs can get into the recipient cell through the exosomes, and they can involve in epigenetic modifications. So what are these epigenetic modifications? So as I said, this microRNA is pretty much small nucleotide chain. It's about 16 to 25 nucleotides. The thing is, they could include, they can have a complementary RNA sequence when compared to another different mRNA, messenger RNA. And what they do is they, we call it sponging. So this mature microRNA, which has the complementary RNA sequence as of the mRNA, it inhibits its function, and it may lead to some chronic diseases. So for example, if there's a mRNA which responsible for fertility and reproduction, if one of these microRNA would bind into its 3' untranslated region, and it would lead to that mRNA's translational repression, the expression of its gene, or it would degrade its mRNA and would lead to chronic diseases. As in this example, it may be a chronic endometritis, or it may lead to poor fertility. So that's the role of this microRNA. So these are pretty much important biomolecules, yet they are pretty much small, and it's not a kind of a spotlight until a decade ago, because everyone were focused on DNA or a gene or a messenger RNA, but now it's been found that all these gene expression is pretty much depend on microRNAs expressed in our body. So in our different stages of our lives, there are different types of microRNAs will be expressed in different levels of our life, like in our adolescence, and even when we are just aging up. All these gene expression is controlled by these microRNAs. And the importance of microRNA in an exosome is pretty much crucial, because as I would go to the next slide, so there are many different circulating microRNAs. Like there are microRNAs free floating in our blood, but when compared to an exosomal microRNAs, these are selectively packaged into an exosomes, and these exosomes have been very tissue originated and very tissue specific, and the microRNAs in each of these exosomes may be different. So the microRNAs in blood or urine or saliva will be different types of microRNAs, and they can be involved in different types of activities and body control mechanisms. And so that's why. So microRNA engulfed in an exosome is pretty much important and critical for our gene expression. And they can be used in different types of ways. So that's the novel researches going on here, because now more researchers are being focused on the epigenetics of life, like not as genetics, because they know now we all we all have 23 chromosomes, and but we have like different types of diseases. We our expression profiles are different. So that is mainly because of the epigenetic modulations. For example, exosomal microRNA are involved in these epigenetic modifications, and we can be used as many other important aspects as well. So I would say, so this is one of the important aspects of exosomes, because they can be used as therapeutics. So we can isolate these exosomes from patient. We can isolate these exosomes from blood or any circulating biofluids like urine or saliva, and we can load any kind of a therapeutic drug into these exosomes. So now the important now many researchers have been conducting the efficacy of these drugs, like efficacy of these drugs have been increased using exosomal therapeutics, because so we use as tablets. So, but these tablets, rather than using it as it is, we can load it into an exosome, and then these exosomes can be loaded back into the patient, and the efficacy of the drug has been severely increased using exosomes, because exosomes would just transport all these drugs directly to the organ or the tissue, rather than just lying around in the blood itself. These are pretty much efficient way of drug delivery vehicles. And yeah, so for example, in cancer therapeutics, now researchers have been conducted to find a way to improve the uh, exosomes as a cancer therapeutics. Like if you have a kind of a DNA, mRNA, or a microRNA, which is related to cancer inhibition of cancer cells, we can just load those biomolecule into an exosome, and then can be treated into the human body, or we can just give it as an oral expression, or any we can just inject into the blood, and those exosomes will travel directly or to the uh, origin of the cancer cells, and then they can easily release the microRNA or this biomolecule into the cells, and the efficacy or the efficiency of the drug will be pretty much increased using these exosomes as a cancer therapeutic. And next, we have found out these exosomal microRNAs involved in inflammation and fertility. So we have found that post and prepartum dairy cow suffers different diseases, for example, endometritis or metritis, which are related to inflammation. But at the same time, we have found out these exosomal microRNAs are expressed in different levels with dairy cows who suffer these endometritis or metritis diseases. So in that way, we have found that there will be we are just conducting our research to identify differential expression profiles of exosomal microRNA between high fertile and low fertile dairy cow. And our plan is to implement those results into human trials, and maybe in different other aspects as well. So, and the other important part is, so these exosomes contain cytokines. So cytokines are they are inflammatory agents, and there are two different types of inflammatory agents, cytokines. So anti-inflammatory and pro-inflammatory cytokines. So as in here, these anti-inflammatory cytokines express in a normal or a very healthy cells, but if there's an inflammation, these pro-inflammatory cytokines expressed. So we have found out these pro-inflammatory cytokines like prostaglandins, IL-8, prostaglandin PGF2 alpha, IL-1 alpha, and IL-8 are highly expressed in low fertile exosomes. But when compared to anti-inflammatory cytokines, they are pretty much lowly expressed in low fertile exosomes. That means there are differential expression of these cytokines in exosomes. So that's interesting. So these exosomes can be used as a diagnostic tool to identify high fertile or low fertility. So, and it can be used in different aspects as well. That's why we are just researching, and many other research groups have been started researchers. And yeah, so that's all the background. So now I would just show you how these exosomes and these extracellular vesicles are isolating in our labs. So the method is pretty simple, but it contains different isolating steps. So the main steps are like differential centrifugation steps. So we have to centrifuge our samples in very high speeds. So the final will be 100,000 G centrifugation for about 2 hours at 4 degrees. And at the end of these steps, we get a extracellular vesicle sample, so which contains different sizes of vesicles, but exosomes also included in it. So then we have to do the exosome enrichment. So we have to we have to select these exosomes from that vast types of vesicles. So here we use size exclusion chromatography. We use a kind of a filter. So it has a kind of a 70 nanometer pore size. So through that, we insert the sample and we collect different fractions. And as in here, so the first fractions will contain larger vesicles, but in the mid-range, we contain these exosomes. And after that, all these small particles and small vesicles, which contains proteins, RNA, will just come out. But fraction 7 to 10, the mid part, will be the main focus, and we will take out these exosomes and just enrich using these QVV columns. And we have to just then we have to prove that these are exosomes, and we are doing these characterizations. So for this, we use 3 different techniques, Western blots. So we have exosomal protein markers, for example, floodling, tumor susceptibility gene, and CD63, CD9. So we use Western blots to characterize exosomes, and we use nanoparticle tracking analysis just to determine the size distribution of the our sample, and we use transmission electron microscopy to confirm the size and the shape of the exosomes as well. So I would show you. So these are transmission electron microscopy images of exosomes, pretty much small. Size is around 100 nanometers. And here also these are NTA, this nanoparticle tracking analysis results. So exosomes ranges from 100 to 30 to 150 nanometers. In here, our samples just have 150 nanometer range, but sometimes we might have extracellular vesicle samples as well. And so these are Flotilene and Flotilene Western blot results. So these bands show that the samples contain exosomes. And after isolating exosomes, we isolate microRNA. So for this, there are different types of methodologies, but we have optimized our using microRNAs in minikit and trisol, which has shown high efficacy levels and efficiency is pretty much higher using these microRNAs in mini columns. And moving on to these, so these microRNA will be sequenced using next generation microRNA sequencing platforms. We use Australian Genome Research Facility for that, and they use Illumina Novasic S1 platform, which uses 100 base pair single and read sequencing method. So from that sequencing results, we can identify different types of microRNAs in samples. As in this example, this is a bovine plasma sample, and we have got microRNA 148A, which is very common in bovine plasma samples. And this is kind of a loop of RNA, but the RNA of interest lies in these red color sequence, which is about 16 nucleotides. And we can identify novel microRNAs, which are the important part. So we identify new and very novel microRNAs in samples, and those are the focus of our research as well. And then we can use these as differential gene expression, like differential expression of microRNAs, as in this example. You can see this microRNA 148, it's highly expressed in these groups compared to these. So in different samples and different animals, the expression levels of microRNA is pretty much different from each other, different from tissue and different from the cell type. So that's the novelty of microRNA exosomal research as well. So yeah, so as in summary, I would just try to wrap up everything here. So exosomes are nanovesicles, pretty much small vesicles, which carries important biomolecules like proteins, microRNAs, RNAs, and even lipid molecules. So my interest is exosome microRNA, which involve in different cellular activities, and I have just showed you the exosomal microRNAs as therapeutics and as a diagnostic tool as well. And finally, I just showed you how these exosomes have been isolating and how we enrich exosomal microRNA. And yeah, I hope we can use this in different ways and different aspects to cure different diseases and use it as a drug delivery mechanism in future. Thank you very much. Any questions?
Thank you very much, Pevindu. That was a very interesting talk, and it's very timely, I think, and bit of technical. So maybe we will get questions today, and we will get questions later as well to whoever's listening to the talk in future. Right. So while we wait for questions, I would like to start with, so in NcodeX, we always promote innovation, right? So what would you say the innovation aspect of this current project?
So the important thing is, like, this is pretty much novel research. So we are trying to find a biomarker for fertility at the end of this research. So we have already optimized exosomal microRNA isolation method, but at the end of this research, we would try to establish a biomarker or or an exploratory biomarker which can identify high fertile or low fertile animals. I mean, we will start it from the dairy cow, but after that we would just try to find it, try to apply for humans or any other aspects as well.
Yeah, that's
Yeah.
great, and something again very technical, but it's has a lot of potential. We have a comment from Vinodya saying it's great talk and extraction of exome is very interesting. And leading from that, I would like to ask, what are the challenges? So you've been working on this in lab. So in practice, what are the challenges you faced when working with microRNAs? Because we will have a lot of young researchers who
That's a
will
good
be getting to this
question,
field.
because microRNAs are pretty much small molecules and they are pretty finicky. So we have to use pretty much sensitive techniques, and I have to go through many steps to confirm that these are microRNAs and exosomal microRNAs. So we have to utilize very sensitive environments, and we have to maintain that environment. That's the main challenge. So, for example, we have to use PC2 laboratories and PC3 laboratories as well when isolating exosomes and microRNAs, and we have to ensure that we maintain optimal temperature. And I mean, if we just use it at room temperature, these microRNAs can degrade. So that's the main challenge. And the other challenge is, so this is pretty much kind of a novel research area, and I'm also learning, and just, I mean, as a research group, we are just learning how to analyze these results, because next generation sequence produce a huge number of results. We have to just filter them out, and it's a vast study area, as in bioinformatics. So that's pretty much challenging, of course. There are different softwares have been introduced day by day, but we have to just select the best software to analyze all these microRNAs and all these sequencing results. That's a pretty much challenge as well.
I can't agree more. It's a lot of noise when working with microRNAs, and then you have to focus on the signal.
Yeah, of course. There are pretty much false signals as well. That's the main challenge. We have to just identify what's the correct microRNA signals and correct microRNA sequences from all these next generation sequencing results. That's quite a challenge.
Absolutely.
Yeah.
Yes, the analysis is a bit of a hectic challenge as always. So I know this is a very new research theme, but you've mentioned cancer therapeutics. And I was wondering, where do they stand at this moment? Are they ready for clinical trials, or are they on their way? If not, how long do you predict they would come out?
Yeah, still they are in animal trial stages. So it would take another couple of years to finally get into these exosomal microRNA, exosomal therapeutics, as in cancer remedials or cure. So, but it's promising. That's the important thing. Like the results are promising, but still we have a large gap of knowledge, and that's why all the researchers are being conducting, and many different groups have been just started off all these exosomal cancer research therapy. So yeah, I think in a couple of years it might just come as an animal trials, and I mean human trials, and it probably after another 3-4 years these drug delivery vesicles will be a huge turning point in cancer therapy, I guess. Yeah.
Absolutely. So 3-4 years is okay when it comes to very long-term scientific research. So in
Yeah,
that
because
case, yeah.
there are many other countries as well. So they also started up all these exosomal cancer research. So probably in a couple of like 4-5 years would be enough. I believe we'll see how it goes.
Yes, let's see. So at NcodeX, we always encourage interdisciplinary research. So do you think any other STEM disciplines like technology, engineering, mathematics, can those disciplines help you and this field get a better outcome? What is your opinion on that?
Yeah. So thing is like these exosomes are pretty much biological, and we have to just sticking to right at this point will be only for biological or any other engineering aspects. But when you talk about interdisciplinary research or interdisciplinary industry related applications, I would say we can use this as in a dairy cow farming model. And so if we can identify high fertile animals in a farm group, and we can select those animals and we can crossbreed them within themselves and increase the high fertile or the high milk producing trait animals, and that will be kind of a way for farmers to identify high fertile animals or high milk producing animals in that group, and they can improve their profitability and improve the quality of life of the animals as well. So that's another way of using this exosomal research into industry. And this can also be used as a fertility biomarker for humans as well at one point. So that would make our human, early identification of our fertility status, and we can improve our quality of life as well. And and there are pretty much different interdisciplinary implications right at this point. But yeah, hopefully it all be evident in next 4-5 years, I hope.
Yes, so that means it's an open invitation to all the STEM researchers to
Of course.
join hands.
Yeah.
Right, so moving on that, so there seem to be a lot of potential applications in that. So where do you think it stands in sustainability, long-term sustainability, especially in low and middle income countries and rural countries around the world? How do you think that could be applicable to your research?
Yeah, so as I said, we can use it as a dairy cow farm model. So if we identify all these high milk producing or high meat producing or any high fertile animal set within a farm animals, so we can improve the food production, and we can improve the quality of life of animals, and that way we can minimize the food wastage. And today we have a global crisis of food. So if we can use these exosomal microRNAs as a diagnostic of those particular areas, like in fertility or any food production, we can use these to improve the food production or a sustainable animal farming techniques. Or we can, and at the same time, we will be like trying to find the efficacy of using these exosomal microRNAs as a biomarker for autism as well. So still autistic kids are being identified after like 5 or 6 years. So if you can use a biomarker to identify these autistic kids, as in way before the birth as well. We can improve the quality of life of the kids. Like we can, from the beginning of their life, we can utilize some developed techniques or tools for their learning and all these their upbringing, and in that way we can improve the quality of life of autistic kids as well. But it's a way more ahead of us, and we have to just sacrifice more time and energy onto this as well. Yeah.
Absolutely. So there's no problem in keeping hopes, and we always need to look far ahead in the next
Yeah,
50
of
years
course.
ahead, maybe.
Of course.
I think we have a few comments as well, agreeing to what you just mentioned. Ara says it'll be beneficial for many, and Prasad says it's beneficial for many countries, and absolutely. So this has a very good potential in sustainability around the world.
Of course, yeah.
So, moving on, what would be the final take-home message from your research you would like to give?
So, yeah, I would say this exosomes and exosomal biomolecules, proteins, microRNAs, or any other lipid, these are kind of a novel research thing, and we have to learn more on these as well, because in a couple of years these will be a turning point for any therapeutics or diagnostics. So probably if anyone can just try to learn, or just to expand their knowledge on these exosomes, that will be really helpful for them for their future careers or future understandings of drugs. And yeah, it'll be really important, the knowledge of these exosomes for anyone, I guess. Yeah.
Awesome. So while we are getting to the end of the Q&A, so Lakshini here will share the feedback form in the comments. By all means, fill it up, give your honest opinion, and any comments, suggestions about the talk and about the program, PhD talks, how would you like to improve the program? And also she will share another form if you know any PhD student, or if for yourself, if you'd like to give another talk, by all means, register at our registration form, fill out the registration form and join with us. Right, so we have a lot of young researchers. Pevindu, finally, what would be your advice for any future researchers in the field, in overall STEM research?
Yeah, I would say the future, like the careers are pretty much challenging, and I know by myself it's pretty much hard to find all these career pathways. But don't give up, and just explore what's in the research area, and just expand your knowledge. And I would say don't give up on anything. And if you need a kind of a if you need to go for a master's or a PhD, or if you need to uh, get into any kind of a career related to research, just go for it and just try it, and you'll be able to achieve what you need in the end. Just don't give up. That's what my message for any young researcher, or any young student.
That would be great, actually. So consistency is the key.
Yeah. Yes.
Right.
And yeah, and thank you very much for giving me this chance to show up my research area, and just to I'm pretty much happy to share my knowledge and knowledge about these exosomes and microRNAs. So and thank you very much, NcodeX, and we'll see you again, probably another meeting, or like another show. Thank you very much.
Absolutely. So thank you very much, Pevindu. I know you're connecting us from Brisbane and it's a bit late in the night. And thank you for your time and efforts. And this is the episode 7, PhD Talks episode 7 on exome-derived microRNA for diagnostics and therapeutics by Pevindu Abeysinghe, who is a second-year PhD student at Child Reproductive Health Research Team, Center for Children's Health Research, Faculty of Health, Queensland University of Technology. So he shared with us his experience today about his knowledge and expertise in epigenetics and exomes in fertility. And it's a very good talk. And if you have missed anything, we will share it in our YouTube channel and in our social media as well. So you can always listen to it. And if you have any questions, by all means, add comments or reach out to NcodeX, and we will by all means link you with Pevindu and he will answer any of your questions. So meantime, NcodeX is an R&D institution. So we are new institution with interdisciplinary research and development, and we have training programs as well. And if you're interested in what we do, please visit www.encodexpert.com, our website, and reach out to us through any of social media, Facebook, LinkedIn, Instagram, Twitter, pretty much anywhere we are there. And yeah, so we will see you next month on the first Saturday for our PhD Talks by NcodeX episode 8. And I thank you very much for joining here with us today, and have a good rest of the weekend. Thank you.